say anything right now.
Okay, we ready to get started? All right, hi everybody. My name is Sue Lana. I'm a medical oncologist here at the Flint Animal Cancer Center, and I'm your moderator today for our panel. Thank you for joining us, and our panel that we are focusing on today is entitled The Answer to Cancer, Start at the Cellular Level, Understanding Foundational Cancer Research. And some of you may know that May is actually National Cancer Research Month, and so we've decided to feature some of our basic scientists and give you a little bit of a insider view into how basic science research helps us treat our patients with cancer. So I'd like to introduce our panelists today. We have Dr. Ann Avery, who is the director of the Clinical Hematology Pathology Laboratory here on campus. We have Dr. Dan Reagan, who's the director of the Comparative Pathology Laboratory, and we have Dr. Samantha Schlemmer, who's the Seeker Oncology Research Fellow in Dr. Doug Tham's lab. So just so you're aware, we are following the COVID protocols that are outlined by CSU, meaning everyone in the room with us that you can't see is masked, and we're separated now by 3 feet or more because guidelines have relaxed a little bit, but we have our masks off so you can see our faces. So we'll get started. We'll go over some basic questions and information with our panelists, and then we'll answer some questions that have been submitted by the audience. And then if we have time, we'll take some questions from the floor. So go ahead and type them into the chat, and then if we can't get to your questions, we'll answer them afterwards by communicating with you directly. So research has always been a big part of the mission at the Flint Animal Cancer Center, specifically comparative oncology research, which benefits all cancer patients, both humans and animals. We have over 10 labs and nearly 100 people who work on this effort. Much of the work is funded by grants that are written by the scientists who we have here in the room, as well as others. And those granting agencies include federally funded granting institutions like the National Cancer Institute, also some of the canine foundations or animal foundations that we work with, like the Morris Animal Foundation. And then we also are fortunate enough to have support from donors, particularly for helping us train our future researchers through our training programs that we have at the Cancer Center. So today we'll be exploring what is basic foundational research and how does it relate to our mission supporting comparative oncology to help both pets and people who have cancer. So we'll have each of our panelists respond to this question. Why is basic science important in our fight against cancer? Ann, do you want to go first?
Sure, I'll go first. So thanks, Sue. Why is basic science important? So I think one of the things that is important to our lab is the study of the genetic basis of cancer. And the kinds of studies that we do are made possible by just dozens of years of basic science research that you would not think would have any relationship to cancer of any kind, animal cancer, human cancer. The sequencing of the human genome followed by the sequencing of the canine genome relied on the efforts of people doing such disparate things as basic chemistry and studying bacteria that live in the wells of the hot springs in Yellowstone. So when you put all those things together, they accumulate to give all of us the ability to do the kind of sophisticated analysis that we do. And in particular, when working with dogs, I would say in the past 5 to 10 years, there has been a logarithmically increased ability to work with domestic animal species because we know so much more about their genes, and we can manipulate, we can do experiments that we couldn't previously do in dogs that in the past we could only do in mice and people, but we now have better tools.
Thank you. So Dr. Reagan, why is basic science important?
Okay, thanks. Yeah, so I'd echo Ann's sentiments really. It's kind of the foundation for all improvements that we see in the clinic, right? So all advancements or improvements in patient outcomes, and kind of similar to Anne's analogy, right? All of these people across the world performing these basic investigations are kind of contributing one word, let's say, to a page in a 1,000-page book, and that 1,000-page book is really what kind of moves the needle on patient outcomes. So I say, without that, you really have nothing. So.
That's pretty cool. 1,000-page book, maybe like a 1,000,000-page book.
Maybe a 1,000,000 pages. Yeah, that'd be more realistic.
It's a Moby Dick kind of thing.
Yeah, that's Moby Dick.
Yeah.
How about you, Dr. Slimmer?
Yeah, I mean, echoing everybody, if I had to summarize it in one sentence, it's basically the fundamental knowledge and understanding of how and why cancer occurs. Then that knowledge and understanding can then be applied and translated to directly patient care to improve prognosis, diagnostics, and treatment.
Great. We'll move on to our next question. We'll start with you, Dan. What role does your lab have in advancing cancer research?
Okay. So yeah, my lab studies cancer metastasis. So kind of how and why cancer spreads throughout the body, and really, the reason we're focused on that is we're quite good at treating primary tumors. So we can surgically remove these tumors. Typically, they're quite responsive to frontline therapies or chemotherapy, and metastasis accounts for kind of 90% of cancer-related mortalities. And so, our approach is, if you can understand those basic mechanisms, again, getting back to that basic science, but understanding those mechanisms of how a tumor cell spreads to a distant site, let's say the lung, for example, and then why, once it's present in this distant site like the lung, that it doesn't respond to our current, therapies that we have, right? So in the primary site it will. In these distant sites, it's really poorly responsive, and that's why we see this high mortality rate. And I think getting that basic understanding of those processes is really what's going to, lead to discovery of novel therapeutic targets and could really kind of change cancer patient, outcomes to kind of managing it like a chronic disease, if you will, something like diabetes.
So how do you do something like that? Do you look at the specific cells, or?
Yeah, so our approach, and this goes back to kind of a hypothesis or a theory postulated by a British surgeon back in the late 1800s, where he kind of retrospectively looked through the medical records of 1,000 women with breast cancer and saw that these tumors didn't necessarily spread to all parts of the body. So there was really distinct locations where the cancer spread to. It was primarily, the bone, the brain, or the lungs, and it didn't go to places like, the spleen or the adrenal gland or basically other highly vascular organs. And so he postulated that it wasn't just something about the tumor cells, what he called the seeds themselves, but it had to be something about those soils, so the same reason that, peaches grow great in Georgia and potatoes in Idaho, there's something about why a breast cancer cell likes to spread and grow in the lung or the brain. And so that's really what we're focused on, is that soil and understanding, what are the cells of that soil? So what's the composition of that soil? How is it being fertilized per se? So how is the tumor influencing that distant soil to make it permissive for its growth or for its spread, and what we hope to gather by understanding that is ways to poison that soil. And so yeah, we take a variety of approaches. We get donor cells from human tissues from sites like the lung, and we can take factors produced by a tumor cell and put those on top of these resident cells of these soils and try to understand how they respond and from an immune perspective, and we feel that if we can understand that response, we could maybe interrupt that response. So and then obviously leveraging the dog as well. So
cool. Dr. Avery, how about you? What does your lab do, and what's its role in advancing cancer research?
So excuse me. My lab specifically concentrates on one set of tumors called lymphoma and leukemia. And these are tumors of lymphocytes, which are, of course, very popular in the world today because they're keeping us all safe from infectious diseases. But sometimes those lymphocytes go bad and become cancerous. And so we're interested in two things. The first thing that we're interested in is how those cancerous lymphocytes are related to the normal lymphocytes that they arose from, and that can give us some clues about what the process might have been between the normal lymphocyte and the cancer lymphocyte that comes from it. And then the other thing that we're really interested in is this really remarkable observation that, so there's many different types of these cancers. Even though we call them lymphoma, there's sort of 50 or so in people and fewer in dogs, but still quite a few. So one of the observations that we've made is that there's really remarkable breed-specific differences in predilection to these different kinds of cancers. So one example would be the boxer breed. There's a particularly aggressive kind of T-cell lymphoma that is 33% of all the cases we see are in boxers. And that's true all through the US, and it's true all through Europe. So there's clearly a breed-specific, which means probably a genetic predilection for that disease. When you have breed-specific predispositions like this, it makes a lot of studies much easier to do than they would be to do in people. So you have a much more closed genetic pool, and so you can look for genes that might predispose to cancer. And then you can also look at these dogs as a group and ask the question, are there any environmental risk factors? Are they exposed to certain things? Sorry, do you need?
Everyone, sorry, I'm going to have you guys pause for just a minute. We're having a connection issue. We are going to reconnect to a better internet connection. Audience, if anybody gets disconnected, you can just join back on the link that was emailed to you and we can invite you back in. So just give us a minute or two to get that fixed and hopefully we'll have a better connection for everybody. Thank you. Okay, everyone, we are back on. So if we can just pick up where we left off. Sorry about the interruption.
That's okay.
That's okay. So thanks everybody for your patience, and hopefully everybody got connected back on. Dr. Avery was just talking about lymphomas in veterinary patients and that there's a big breed predilection, particularly with boxers and T-cell lymphoma. So off you go.
Okay. So anyway, the sort of purpose of discussing that is in human medicine, if people want to understand the causes of cancer, so in order to create preventions, which of course are much more efficacious than treatment in the long run, you have to do these large epidemiological studies. So things like the Framingham Heart Study and the Nurses' Health Study are why we know that smoking is bad for you and diet can cause heart disease. You can do those kinds of studies in dogs with a much smaller population, because if you look at a single breed, you have a less genetically heterogeneous group. And so it's much easier to identify risk factors. And we can talk about the lifetime study later on. But so we're interested in, now that we've identified these predilections, we're interested in using those observations to look for things that we can prevent in a dog's history that might help mitigate the development of cancer.
A follow-up question for you. You had mentioned that in humans there's over 50 different types of lymphoma. As a medical oncologist, I don't think we recognize that many in dogs. Is it because they don't have them, or we just haven't found them?
Right, so it's not because they don't have them, it's because we have, up until recently, lacked the tools to differentiate different subtypes. There's probably a dozen well recognized in dogs. The other thing, again, getting back to the breed specific question, is there may be some tumor types in people that are the result of the particular configuration of the human genome that we probably will never see in dogs, or see only rarely, because the dogs have a different genetic configuration, and so they're not predisposed.
Great, thank you. Dr. Schlemmer, how about you? What does your lab do in advancing cancer research?
So a lot of what we focus on is targeted therapeutics, and so sort of like precision medicine, right? And so with that concept, we're trying to target specific genes or proteins that are either expressed or present within these cancer cells. And so by having drugs that directly impede that, then we can stop, hopefully, the cancer from growing. But generally what these proteins do, and if you look sort of, we'll use HER2, or human epidermal growth factor receptor 2, as an example. So back in the 80s, they found that this specific protein receptor was amplified on a lot of human breast cancer samples. And so that led to the development of a targeted therapeutic called a monoclonal antibody, trastuzumab. And so that directly targets that specific receptor from signaling into the cell, wanting it to proliferate and signal and continue to invade and metastasize. So ideally what we're trying to do is identify what these specific genes and proteins are in tumor cells that are aberrant. So whether they are mutated or overexpressed or amplified, can we target that specifically so that it stops signaling? But the problem with that is that cancer is very adept at mutating. And so a lot of times when we do find a specific pathway or protein that is mutated, the cell can also develop other mechanisms to signal to proliferate and invade. So a lot of times, at least now in human medicine, they use multimodal therapy. So in addition to classic chemotherapy, which is cytotoxic and doesn't really specifically target cancer cells in general, it targets the rapidly proliferating cells, and so those could be present in your GI tract or potentially your bone marrow. But with these targeted therapeutics, they can specifically latch on to those cancer cells and mostly save the normal tissue around it. So using that in combination, they've seen better effects long term.
Awesome. And it sounds really complicated to me. Is it really complicated?
It is, yeah. And I wanted to just show just exactly the different pathways that could be present. So this is just a little poster from one of our cancer.
All of us have one of these in our lab.
One of our cancer books, and every lab sort of has this, and I don't know how well it'll show up on the virtual, but there's just so many different receptors that are on the cell, and pathways that can signal into the nucleus for the cell to proliferate and grow. And so it's not just necessarily one thing that is aberrant in a tumor, it's usually multiple things. And if it's not multiple things and we target one thing, then it sort of will compensate because it wants to survive and proliferate. So yeah, it seems easy, and it's like, oh, if we could find just that one mutation to cure cancer, like we could do it. But these cells are just so complex and they just want to survive. So it's by finding these mechanisms and again, targeting potentially different pathways and using multimodal therapy that we could potentially get better outcomes.
Awesome. So there's no magic bullet.
No, I wish.
Yeah, I wish
We're
too.
all looking for it.
So along those lines, I'll go back to Dr. Reagan about this. One of the big areas of advancement or research in human medicine as well as in veterinary medicine is harnessing the immune system. And so can you talk a little bit about what your lab is doing with that?
Yeah, absolutely. So some people may think that may in fact be the magic bullet, but
Like you, for example.
Yeah, like me. No, no, it's not clearly. But yeah, I think it comes back to the fact that your immune system is essentially designed to eliminate cancer cells and has co-evolved in your body to eliminate these transformed tumor cells, just as it does the virus of the common cold, right? So unfortunately, in some people, it becomes dysfunctional, and that's when it's no longer good at eliminating tumor cells, and these clinically detectable tumor masses arise. And so, I kind of look at it as, why not just fix the roof instead of build a brand new house, right? So, I think with the goal of immunotherapy, right, is to kind of reinvigorate or reestablish that normal anti-tumor immune response. So to get those lymphocytes that Ann was talking about, the most important effector immune cell probably for cancer immunology is the T lymphocyte, right? So get those guys working again, get them killing tumor cells, and again, it's much better to restart that process versus invent the wheel. And so, from my perspective, again, we're really trying to target the soil. So we're trying to block some of those tumor promoting immune processes that are going on in these distant sites to slow or prevent metastasis. But other strategies are to, again, to kind of reinvigorate those T cells. So you can give targeted therapeutics like Sam was just talking about. So antibodies that can bind the surface of that T cell and give it an activating signal that can kind of turn it on and put the foot on the gas and get it ramped up to start killing tumor cells. And the way that these cells of your immune system recognize the cancer is through antigens or tumor antigens, which are kind of signals that this is a foreign cell, if you will. And so, other strategies that people are investigating and we've looked at in the past is delivering vaccines, which is kind of like a bulk dose of these antigens to kind of get your immune system to wake up and say, hey, there's something foreign here. So, I'd say we're taking a combination of approaches, and just like Sam said, it's going to take that kind of multimodal approach to maybe awaken these T cells, modulate the soil to, a less tumor promoting or immune suppressive type soil, and kind of tip the scales back to where they originally were, right? Where your immune system was really effective at eliminating these tumor cells, just like the common cold.
Great, thanks. We'll talk next a little bit about some questions that had been previously sent in. And so and this will be for all of you to answer in turn. In the next 2 to 3 years, what research discovery do you think will have the biggest impact on how we treat cancer patients, either veterinary, human, both? Doesn't have to be in your lab, but it sure could be. That'd be cool.
Sure hope so.
No. Yeah, I sure hope so. So Sam, do you have anything? No?
No.
It's okay. You're thinking. We'll let you think about it. How about you, Anne?
My feeling about cancer research is that it's incremental. I think everybody agrees with that. For example, I used to be a big skeptic about how the immune system would be effective against cancer, but people like Dan and Steve have proved me wrong through incremental advances. So I think probably the main set of incremental advances that we'll see over the next couple of years is a better understanding of the mutations that are present in cancer, and particularly, and I think this is pursuant to what you said, how those mutations interact. And so eventually, through very sophisticated data mining and bioinformatics, we might be able to say, we know everything about the DNA in this particular tumor, and we can predict what we can treat it with. I'll just put in a plug, though, also for research into prevention, which I think is another thing that now that we have this ability to mine a ton of data, I think we will find more and more things that we can do to prevent cancer that we were unaware of before.
How about you, Dan?
Again, I think Anne's kind of right on target right there. I mean, prevention would definitely be key. But with the advent of some of these technological advances, I think we're down to kind of single cell resolution, both at genomics, proteomics, so studying these actual signaling proteins. Every process of the tumor cell, we can look at it at single cell resolution, and so that really captures that heterogeneity of the tumor and all that chaos that's going on that kind of Sam talked about. And like Anne said, kind of gives you that blueprint where you can start to figure out how to take that multimodal approach to kind of cover that whole dysfunction, if you will.
So if we have a bigger understanding of what's going on with each particular cell and its environment, how do you see that translating into how do I change how I get treated if I get cancer or I treat my pet with cancer? How do we make that leap?
Yeah, great. So I mean, it's this personalized medicine we've heard about, and then it's even, I guess, a more granular approach to that where you're treating kind of multiple aspects of that tumor, right? So you're going to need to use therapy A because 33% of the cells seem to have this kind of activation going on, and then we also got to layer on therapy B because another third of the tumor is really relying on this mechanism, and then we know that after you give these two therapies, this new population is going to show up with relying on here, and so we're going to have to then hit them with therapy C, kind of. So it's really going to tailor that to everybody in a more granular way than just kind of surveying the whole tumor to see what's most prevalent, but we're going to figure out everything that's kind of at work to promote this tumor's growth.
Can I add one
Yeah,
thing?
yeah.
So one of the other things that a lot of people are working on now is early diagnostics. And I think when we're still relying on some bulky change in the body, like a tumor or a mass or some change in the blood. And a lot of companies now and research is focused on early detection. And I wonder if maybe our entire approach to cancer is going to change when we can detect it when it hasn't metastasized yet, and when it's just a little tiny, just a little tiny whiff of a cancer. The way we treat that might be completely different, and so very sensitive diagnostics may also be a big breakthrough.
Anything to add, Sam? Nope. We're relying on you when it gets really complicated and the cells figure it out to stop them again. So I'll go to another question, which we often think about, but what's your biggest barrier to making progress in your current work? So in what you're doing, what's the thing that slows you down? So Sam, do you want to take that one?
At least for me, as a trainee in a fellowship, it's always time. I expect things to progress a little bit quicker, and science is science and cancer is cancer, and so things don't always go as quickly as I would hope or plan to, and it's just a trial and error process, and that's what basic science is, right? We find out 99 ways of how not to kill cancer, and then maybe the one way that we can kill it, and so yeah, it's definitely time would probably be a big thing for me.
How about you, Dan?
Definitely time, and Ann said that, it's kind of so incremental, right? And then, money, of course. I mean, these with this increasing technology is kind of increasing cost to conduct the experiments that you really need to do to make those incremental discoveries. And so, money's kind of always a limiting factor, I'd say.
How about you?
So yes, I can add to that. I think one of the things we all could use is people who are interested in veterinary medicine now, who are trained in bioinformatics, because that's just a world that none of us, I don't mean to make assumptions about you guys, but none of us are we're all trying. And so I think one place that if we were going to increase funding for would be for training veterinarians in this current world of bioinformatics and data mining and things like that.
Yeah, I think just as a comment on that, with learning more and more about the individual cell and all the different pieces that go into all the different pathways, all the different genetic changes, it's so much information that somebody has to understand and look at and see how what's important, what's not. And I think that's one thing that we've all noticed that is difficult, is figuring out who can look at that information, because we know that, as a veterinarian, I'm not trained to do that. And so I would have to become a specialist in that particular area. And I think that's what we're lacking too.
Yeah.
So good point.
Yeah. I mean, just to add something to that, I think, a lot of people that do have that training sometimes kind of gravitate towards the human field, either just due to an unawareness of comparative oncology. And so, kind of to Ann's point, getting that money, it almost has to be a homegrown or grassroots approach to train these individuals. And so kind of, doing it from the bottom up here within veterinary medicine and having the funds to do that would be key.
Yeah. Anything to add, Sam? You're finishing your training program now.
I am, yes. 2 years, yeah. And definitely the pandemic definitely set things back for us for a little bit, because we were on lockdown for a few months. But yeah, we're back and running, so I'm trying to get as much data as I can in the next couple of weeks.
Awesome. We'll go ahead and take a couple more questions that had been sent in, and we've received several questions on the topic of prevention of cancer. And so we're going to kind of lump them together. They revolved around nutrition, supplements, household chemicals like Roundup. So I'll kind of combine them all into one question. But what do we know about preventing cancer in companion animals, and what efforts are underway to learn more? So Ann, you want to start this one?
I can start, yeah. So I can first just talk about, again, I have sort of said this already, but the value of epidemiology and epidemiological studies looking at prevention. So there's an epidemiologist called Larry Glickman. Can't remember what university he's with, but he did a study in about 10 years ago looking at dietary interventions, or looking at the diet of Scottish Terriers, I think, who are a breed that get bladder cancer. And what he found was that if their owners fed these dogs carrots, and I'm not making this up, they reduced the incidence of bladder cancer by 70%. That's amazing, and all you have to do is buy carrots. Unless you go to Whole Foods, they're kind of expensive, but I mean, it's a very inexpensive intervention for prevention. And that kind of study, which was a pretty small study, I think illustrates how larger studies can really identify many different things that we can do prevention-wise. So right now there are 2 large-scale studies that are similar to the human Framingham Heart Study, if you've ever heard of that, or the Nurses Health Study. Those were studies, I can't even remember when the Framingham Heart Study was initiated. I think it was in the 50s, which followed a group of physicians for a long time and took yearly information from them, as well as blood and tissues and things, about what their diet was like, what their exercise was like. And through that study, they discovered things like what are predisposing factors to heart disease? Why does the relationship between smoking and lung cancer? So right now, the Morris Animal Foundation has and many of you guys probably know about this, has an ongoing study called the Golden Retriever Lifetime Study, which is a smaller study. It's only about 3,000 dogs, but it's huge still for veterinary medicine, which is aimed at looking at those same questions. So the dogs enrolled in this study fill out questionnaires each year, they give samples each year, and the specific goal is to understand the risk factors, both environmental, like what do the dogs do, what do they eat, as well as genetic, what kind of genes do they have, that predispose dogs to a couple of common tumors in Goldens. But we will get a lot more information than just those 3 tumors through this study. The other study is called the Canine Aging Study, and that's funded through the National Institutes of Health, and that's even bigger. That's a huge cohort of dogs, any breed, and those dogs are going to be followed over a period of years, and there's a subset that are being treated with a drug that is supposed to mitigate some of the effects of aging. And I know less about that study, but that's another one that's in this same model to look at preventative therapies.
Yeah, I think the Canine Aging Study is following the patients or the enrollees over 10 years. And largely, to begin with, it'll be survey-based, is my understanding, when patients sign up. And then they'll follow them, gather information, and then depending on the information you provide, they may shuttle you into a sub-study for specific things like heart disease or other types of things. And because it is a study looking at older patients over their course of their lifetime, cancer will be one of the diseases that ends up occurring during that period of time. And so gaining more information about the characteristics of those patients will be important, I think, to learn, like they have in humans, different things that are associated with cancer. So how about you, Dan? Do you have any prevention info?
I don't. I mean, I think that was a great summary. I mean, I guess I'd just point out that the kind of comparative approach is the real value too, right? We're going to reach these study endpoints so much faster than we would in a human population, and gather so much information so much more quickly. So,
yeah. How about you, Sam?
I just know that here at CSU, as well as Wisconsin, Davis, and I think Arizona, are looking at the vaccine against canine cancer too. You could probably speak more to that because you're on the clinic floor, but that's one of the studies that is ongoing that could have information about cancer prevention in dogs.
And that study, in previous panels that we've had, Dr. Tham has talked about that study a little bit, and my dog's enrolled in that study, but it follows them as you have to be a certain age to enter, the age that cancer occurs, and then they follow them for up to 5 years. Some are treated with an anti-cancer vaccine, which is the novel investigated agent. The others are not. And so we'll learn a lot about those patients as well as they go through. And they collect samples of blood and then tumor tissue when it occurs. And we'll have a whole bunch of things that Anne and Dan and Sam can analyze over the course of time and learn a lot from those. So those are the types of things that are happening on the prevention front, I think. Another question that has come up is concerning nutrition and food and things surrounding patients who have cancer and undergoing treatment, are there any recommendations that we have for that? And I actually reached out to our nutrition specialist at CSU and asked her kind of what her thoughts were on that subject. And I think there a bunch of different things came out of that conversation, and I'll send out some follow-up, or I'll have Tori send out some follow-up information for people who are on this webinar today about that response, and it's some really very useful information. I think her bottom line was, and we've learned this very recently in veterinary medicine, is that having a complete and balanced diet is very important versus just specific things. Many of you may know that dietary-related dilated cardiomyopathy is something that we've seen in our veterinary canine patients in the last few years, secondary to certain types of diets. And so nobody knew that was going to happen. They started giving different diets to dogs that were a little bit outside of the commercial spectrum, and then we have this disease that has become a big problem. And so I think those things are going to be very important to understand what it is you're giving, and working with a nutritionist, a veterinary nutritionist, is really important too. So those are all the questions I have. Do you have questions from the floor, Tori?
We do have a couple. So the first question is, do any of you have a story to tell about how basic research work created the foundation into a treatment for dogs or even humans?
Treatment is not my
You got one?
Sure, yeah. So yeah, I did my PhD here at the Flint Animal Cancer Center in the lab of Steve Dow, and so we, again, going back to looking at the soil and the immune system, we took a blood pressure medication that is taken by millions of Americans. It's called losartan or Cozar, and it's used to lower your blood pressure. And we, through basic science, benchtop work, looked at how this drug could block a population of immune cells called monocytes. And again, we started, in a plastic dish, if you will. We moved that into mouse models that we use to study metastasis, and we showed that if you gave this blood pressure drug and gave it at a little bit higher doses than you normally would, you could block these monocytes, and when you blocked them, you slowed tumor spread to the lungs. And so that data was kind of enough in the mice, was enough to move that drug into treating dogs in the clinic, just right downstairs below the lab. And treating dogs with bone cancer, which is a cancer that has a really high propensity to spread to the lungs. And so when we did that and we kind of escalated the dose of that drug, we were again in these dogs able to block this immune cell population, and we were able to see some responses in dogs that kind of had end stage, lung metastatic bone cancer. And so those responses again were kind of enough to now take this therapy down the road to Children's Hospital Colorado, and now we've also opened up the trial for enrollment at Children's Hospital Atlanta, and so now we've moved this therapy into the human clinic treating children with bone cancer, and again, we're really early in that trial. We're at that low dose, and we really feel like we need to again escalate that dose to that higher dose. But I think if we saw just the same level of response we saw in the dogs and these kids, it'd be a huge improvement. So
that's a great example of seeing a discovery in your lab on the bench and then moving it forward.
Yeah,
I remember
think,
those patients.
Yeah, and I think that's what everybody at the ACC is really trying to do. When whether you're on the really early side making those basic discoveries where that allows somebody to build on that to then try and translate a therapy into the clinic. I mean, that's always the goal in the end. So
how long was that process?
Yeah, it was
How old are you now?
Yeah.
I know,
Throughout
where
your
did time
whole
go?
PhD.
I'm about ready to retire, so no. Like 2012 really started heavily, and the trial in kids opened in 2019. So it was like 7.5 years. So yeah, and I just actually finished the revisions for the paper to submit the paper, resubmit the paper for the dog work yesterday. So
Great.
So it's a long process.
But probably a quicker process than had you not done the work in dogs to try to even move things into people.
Yeah, absolutely. I mean, 7.5 years, and this is we're repurposing a drug, which is a huge help. But I think that the stat that's thrown around is it's typically about 16 years, and I guess testing it in dogs really gives us a kind of a signal of hope that the way it worked in dogs, it provides more predictability than just a mouse, if you will. Yeah.
Mm-hmm. Great.
Yeah.
The next question we have is, how will we know if we have a small tumor so that we can stop it from spreading before we see a doctor and find out we have cancer?
So I think that question speaks to what you were talking about, Anne, that diagnostics and how can they help us?
Yeah, so to a certain extent, it's going to be tumor specific. There's ways to detect lymphoma, which we're interested in, and that's going to be different than ways to detect maybe bladder cancer. But one of the focuses currently in diagnostics in people is just taking a blood sample and then sequencing all the DNA in that blood sample and determining if there are mutations associated with cancer, which would indicate that somewhere there is cancer. I think the thing we all need to be careful of is direct translation of that to dogs. So we need to be sure that we can demonstrate that those kinds of diagnostics work for our species as well. That's going to be a lot of work, but eventually I think 5 to 10 years, we would also be able in dogs to have that kind of very sensitive diagnostic.
Another question, this person has heard there is a lifetime study on Goldens. How can that study benefit Goldens, all dogs, and even humans?
Do you want to talk about that again?
I can speak to that briefly. Right, so that study, I think Dan made a really good point, which I neglected to state. So that study is going to follow dogs from the age of 2 until the end of their natural lifespan, which in a golden retriever is probably 13 years old. And so we can, unlike a human study where we might start at the age of 2 and follow an individual into their 70s or 80s, we can learn an enormous amount about the process of aging, the predisposing factors to cancer and other diseases of older dogs. And so I think without question that study will give us information that we could not get on a scale on any human study of that size and for that cost. I mean, it's still, I don't know how much this study is. It's $30 million, I think, something like that.
Probably, but it's probably a fraction of the cost to do a similar study in people.
Right, tiny amounts of money compared to what a human study would look like. And so I think one thing unrelated to cancer, one of the processes that we can study in the golden retriever lifetime study is the process of aging and the changes that are, the genetic changes that are associated, the changes in your blood chemistries and things like that, and how those might be mitigated as well.
Yeah, and I think that with the data from this study, since these dogs are living in our same environment, even just realizing environmental factors that could be at play. And so that's why they're a good model for us to study as a translation into human medicine as well.
Yeah, that's a really good point. I think one of the other things that we will end up learning from a study like this, just like they did with the nurses study, the Framingham study, is that you have your data set and it keeps being looked at and mined and going back and looking at things. So as we learn more and more about the genetics of this particular disease, we can go back and look at the patients and the samples from the patients who are enrolled in that study 20 years from now, because all those samples are banked and all that information is saved, and continue to learn more. So they'll have some primary endpoints that they'll be able to make a lot of conclusions about and learn a lot about, but in the future, that's where additional value comes from studies like this, is being able to go back and say, hey, we didn't know smoking caused this, but wait, smoking does cause this. And now, and that's what they learned from the human side of things from those studies that were done so long ago. So it's kind of the gift that keeps on giving, I think, for dogs of all breeds, not just golden retrievers.
Yeah, can I offer just one specific example in terms of the aging question? So recently, and this is pretty recent data in people, people have been observing the fact that as human individuals age, we start to accumulate mutations in our DNA that are similar to the mutations we see in cancer. But this is happening to people whether or not they have cancer. So I probably have a lot more of those mutations than Dan or Sam, but I still don't have cancer. So we think that this also happens in dogs, and the question in people is, what is the effect of those mutations? Are they doing something else? Will those people eventually get cancer? We can study this in golden retrievers and within 7 years learn the answer to that question, whereas in a human study, it's going to take 30, 40, 50 years to find that answer. And that's a really very popular investigation right now.
And I think the other thing that brings up is, perhaps with those people who have that identified trait, why don't they get cancer? What's different about their immune system, perhaps, or what's different about their pathways that makes them more resistant compared to somebody who might actually get the disease in question? And so I think sometimes basic science or some of these studies bring up more questions than we have answers to, but that's all part of research and science is seeing what we learned and then what didn't we learn and what else do we need to ask and what additional questions do we need to ask.
A two-part question. So I'm going to ask the first part for all of you, and then the second part is for one person in particular. Training veterinarians to do basic science is a big need, as many of you have said. What was the inspiration for each of you to do this work?
Who wants to go first?
Sam, you're the youngest. You go ahead.
I guess I'm the actual trainee.
You're the trainee.
Wow. I guess for me, I'm actually a veterinary clinical pathologist by training, but I guess during veterinary school, they offer a summer research fellowship program through the NIH. It used to be Mariel back in the day, but I think it's NIH nowadays. And so I got a taste of basic science with an oncologist at the University of Florida, and that sort of sparked my interest in oncology and pathology. And when I finished my residency, I just wanted more training, and this fellowship was new, and it's one of its kind, really, creating these clinician scientists in the veterinary field. And so I just jumped on it because I really like studying cancer and being able to help patients, maybe not right next to them on the clinic floor. I'm back in the pathology lab, but yeah, my main motivation is to help the clinicians and ultimately help the patients and their owners as well, to have peace of mind with a diagnosis and a prognosis and a good treatment plan. So,
great. How about you, Dan?
Yeah. What was the question? How I ended up here?
How'd you get here? What inspires you to do what you do?
I mean, to be honest, I entered vet school, I guess totally unaware of the opportunities in veterinary medicine and completely unfamiliar with research. I had only kind of limited clinical research experience in undergraduate education, so I wanted to just own a practice and treat animals and help animals. And when I got into vet school, I realized that we don't know everything, and everything's not written. I know. I told you I was really naive. Like everything's not written down in a book. So they teach me something, and they'd say, yeah, horses or dogs get this, and we don't know why. And I'm like, wait, what? Like, well, somebody needs to figure that out. So I, that was like my passion to become a pathologist, was studying mechanisms of disease, and I realized that research goes, basic science research goes hand in hand with that. And that was really my interest, and I became interested in immunology because it was so fundamental to the mechanisms of a variety of different diseases. And then my own father got cancer, and that really drove me towards cancer immunology and to kind of focus down on that. So yeah.
How about you?
I don't have a particularly inspiring story. I did my PhD in the still obscure area of equine reproductive immunology first before I went to vet school. And then I went to vet school and discovered, I wanted to go to vet school because I wanted to have a better understanding of the whole organism that I was working with as a PhD student. And then I discovered that I just loved medicine because it was a puzzle to figure out. And so I just thought working up cases and treating animals was just intrinsically fascinating. And ended up here, not specifically driven by cancer, but that's just because sometimes that's where your research takes you to places that you would not have necessarily predicted.
Yeah, cool. What's part two?
Follow-up to that question is specifically for Dr. Schlemmer, who is part of a training program. Why do you think this training program is important, and what do you hope to do when you're finished?
That's a great question. And I do know what I'm going to do. I didn't know a few weeks ago, but I do know.
Great.
So I think it's really important. Like I said, there's not a lot of programs out there that teach veterinarians how to be scientists unless they go through like a PhD program, either like in Anne's case, before veterinary school. There's some combined DVM PhD programs, or potentially people will revisit it during their residency or after their residency to do a full PhD. And those can vary in the length of time that you would need to complete a PhD. And for me, for this fellowship, after I've done, I did some years in practice and then my residency, and I didn't have the stamina to do a full PhD program. That was going to take several years, and this fellowship was 2 years with all the rigors of a PhD program. And so I'm getting to learn how to do basic science. I'm doing hypothesis-driven research, and so I'm asking question and answering that question. And I think that that's important because, again, like I said, there's just not very many veterinarians out there that have that opportunity, and so I felt that that was important. And for me, I'm actually going to be joining the faculty at the University of Georgia as a clinical pathologist, but part of my responsibilities as a faculty member will be to do collaborative and independent research, and so I plan to continue to do some oncology research there too. So,
do you feel like your training program has set you up for success?
I do, and I think it's given me a leg up in the application process. I think it makes me different and unique because I don't think anyone else that has done something like a research fellowship before, and so yeah, it definitely is something that I could offer that I wouldn't have been able to offer straight out of residency.
Great. Any more questions, or are we almost done?
I've got one more, I think. Is there a place that an individual who is about to acquire a puppy could go in order to find out if there are any clinical lifetime trials or studies on their breed, similar to the Golden Retriever lifetime study?
That's a great question.
I know the AVMA website, I think, lists clinical trials that are searchable by the public.
Yeah, so what Sam's talking about is the AVMA has a website that lists all the clinical trials that are inputted in veterinary medicine into that site. From a standpoint of someone who's acquiring a puppy, the American Kennel Club, Canine Health Foundation, if it's a purebred puppy, is often a place where people will go to find information like that. The Golden Retriever lifetime study is done entering patients. The canine aging study is still open for patients. I don't know how old you have to be, if they'll accept puppies under a year or not, but their website's really nice and easy to navigate, and so that's probably where I'd start.
Got today. There are congratulations pouring in for Dr. Schlemmer, so wanted to pass that along. And that's all the questions we have.
Okay, so to wrap things up, thanks everybody for joining us today. If you do have any questions that are submitted after the fact, Tori will shuttle them appropriately and we'll try to get them answered as best we can. She's also going to send out a link to some videos that we've produced this month, looking at the different laboratories within the cancer center, and they're short, they're featured on our YouTube channel, and they kind of explain from the investigator's point of view and some of the other folks within the lab what the labs do and why they do what they do. And so they're really nice introductions to some of the basic research labs in the cancer center. So thanks again for joining us, and we'll go ahead and sign off.
Thanks, everyone.
Thank you.